Hi, everyone. Welcome back. This is Jonathan Cedillo, student at Grand Canyon University. And today I'm going to be talking about the blood brain barrier. Presentation for the class Biology 515 Concepts of Human Physiology 1, Professor Sandra Barrera. The blood brain barrier, or BBB, is a boundary between substances, proteins, and molecules present in the central nervous system. This barrier is permeable, which function is to keep the normal regulation of the brain. Brain endothelial cells are the main component of the BBB. These cells have duties to fulfill, such as paracellular transport. Furthermore, neuroinflammation can be caused by factors such as tumor necrosis factor alpha or TNF alpha and interleukin 1 beta or IL-1 beta, which can have negative effects on the regulation of the BBB. Interestingly, this phenomenon can potentially lead to Alzheimer's disease, or AD, due to the aggregation of amyloid beta peptide. In the following article, TNF alpha and IL-1 beta modulate blood-brain barrier permeability and decrease amyloid beta peptide efflux in a human blood-brain barrier model by Romain Bercel et al. An investigation that reveals if the TNF alpha and IL-1 beta change the selectivity of the blood-brain barrier and disruption of amyloid beta trafficking across the brain endothelial cells. During the experimentation phase of this study, human BBB in vitro was used. This experiment allowed the demonstration of the alterations that TNF alpha and IL-1 beta induce towards the brain endothelial cell selectivity. It changes in the organization of adjacent cells and the effect in the regulation of transcellular transport. In addition, the paper also reveals that tumor necrosis factor alpha and interleukin 1 beta that regulate the synthesis of genes LRP1 and BCRP, together with the export of amyloid beta peptide. After gathering the data, scientists were able to decipher the mechanisms of the central nervous system inflammation in correlation with the BBB selectivity and benefit the amyloid beta segregation. In the article discussed, scientists induce an inflammatory response by adding TNF alpha and IL-1 beta to the BBB model. An MTT test was used to measure the number of healthy cells. Dimethyl sulfoxide, or DMSO, at 10% was used to measure cell toxicity. Figure 1 right here and 1A demonstrates that after 24 hours of DMSO exposure, cells were reduced by 26.5% in comparison to the control group. On the other hand, exposure to TNF alpha and IL-1 beta did not show toxicity effects on the brain endothelial cells. Continuing, inflammatory activity is regulated by adhesion molecules such as intracellular adhesion molecule 1 or ICAM1, vascular cell adhesion molecule 1 or VCAM1, and the inflammasome molecule not like receptor family, pyrine domain containing 3 or NLRP3. Using Western immunoblood assays, scientists evaluated the activity of these inflammatory molecules on the brain endothelial cells. They revealed that proteins levels of ICAM1, VCAM1, and NLRP3 were low in the control group. However, higher in presence of tumor necrosis factor alpha and interleukin 1 beta, as shown in figure 1b. But TNF alpha showed a more aggressive inflammatory response than IL-1 beta in the presence of inflammatory factors of ICAM1 and VCAM1 and NLRP3, also shown in figure 1b. In figure 1c, we can see that the expression of ICAM1 is higher in the presence of IL-1 beta. This is only suggesting that pro-inflammatory cytokine treatments do not affect the health of cells. However, they do induce inflammation in brain endothelial cells. Interestingly, according to the article of interest, this inflammation in the brain endothelial cells show upregulation in the paracellular and transcellular transport of molecules across the BBB. Modification in cellular junctions leads the way of amyloid beta. The permeability of the blood-brain barrier is due to the properties of tight junctions and adherent junctions in the brain endothelial complex. According to the authors of the paper presented, it appears that the presence of TNF alpha and IL-1 beta has a negative effect on the expression of these junctions. In addition, this is also suggesting that the presence of pro-inflammatory molecules and irregularities in the junctional complexes allow disorders in the trafficking of amyloid beta, which is an indicative factor of Alzheimer's disease if it's segregated. TNF alpha and IL-1 beta factors effect in amyloid beta transporters. According to the paper discussed, in the presence of TNF alpha and IL-1 beta, proteins LRP1 and BCRP are downregulated to almost 50% of their normal concentration. These are critical proteins that are involved in the amyloid beta peptide transport. In addition, the phosphatidylinositol binding clathrin assembly protein, or PICALM, is importantly involved with protein LRP1, which is also another factor that plays a role in the amyloid beta peptide efflux from the brain to the vascular system. However, PICALM expression is mostly upregulated by IL-1 beta, while it appears that TNF alpha has no effect on the regulation of this protein. This information is suggesting that TNF alpha and IL-1 beta closely regulate the expression of proteins BCRP, LRP1, and PICALM in the brain endothelial tissue. Transcytosis of amyloid beta peptide. We already covered that TNF alpha and IL-1 beta regulate the synthesis of proteins LRP1, BCRP, and PICALM, which have a role in the efflux of amyloid beta peptide through the brain endothelial tissue. In the article, scientists exposed amyloid beta to pro-inflammatory factors to analyze the influx and efflux of amyloid beta after FITC inulin was added. The amyloid beta 140 peptide was used for all experimentation due to its high abundance in the brain. FITC inulin is a marker to trace the transcytosis of amyloid beta. After the addition of pro-inflammatory cytokines, transcytosis in the efflux was considerably reduced in comparison to the control, as shown in Figure 2. From the 100% transcytosis set by the control, the efflux of the amyloid beta peptide was reduced by approximately 26.9% when TNF alpha was present and only 25.9% in the presence of IL-1 beta across the brain endothelial cells. While the influx of the peptide was not significantly reduced in the presence of the cytokines, this only suggests that AV peptide or amyloid beta peptide will be accumulated across the brain endothelial tissue due to the improper exportation of this same molecule. Finally, to conclude with this presentation, the regulation of the blood-brain barrier plays a critical role in the central nervous system's homeostasis. Disruptions in the regulation of the BBB can bring negative effects that can lead to diseases such as Alzheimer's. The presence of pro-inflammatory cytokines, TNF alpha and IL-1 beta, can cause irregularities in the BBB selectivity by affecting paracellular and transcellular transports. TNF alpha appears to increase the selectivity of the BBB in the paracellular pathway, while IL-1 beta seems to mostly affect transcellular transport. These factors attack the brain endothelial cells by downregulating the efflux of amyloid beta peptide, since the expression of proteins LRP1 and BCRP is downregulated as well. The segregation of